Hey guys, my name is Didi Jimenez, and today we're going to go over water and urea reabsorption. First, we're going to start with water reabsorption, which occurs throughout the entire length of the tubule, where water is passively reabsorbed following actively reabsorbed sodium. Interestingly enough, of the water filtered, 65%, which is about 117 liters a day, is reabsorbed in the proximal tubule. Adding on to this, we have another 15% that is reabsorbed in the loop of Henle. Now, reabsorption of this 80% mentioned happens in any condition, aka it's unavoidable. The other 20% is reabsorbed in the distal part of the tubule and is controlled by hormones depending on how hydrated the body is. Aquaporins, aka water channels, are the main form of transport for H2O during reabsorption. These channels are formed when specific plasma membrane proteins are in tubular cells. With this being said, throughout the nephron, there are different water channels. For example, in proximal tubules, they're called AQP1 and are always open, making them accountable for water that is highly permeable in this area. On the other hand, we have the channels in the distal parts of the nephron that are called AQP2 channels in principal cells that are regulated by vasopressin, a hormone. Most importantly, in the proximal tubule, the main driving force for the reabsorption of water is a compartment of hypertonicity in the lateral spaces between the tubular cells made by the basolateral pump's active extrusion of sodium. The concentration of sodium decreases rapidly in the tubular fluid and tubular cells due to this pump activity. At the same time, concentration increases in the localized region within the latter space. Water passive net flow from the lumen into the lateral spaces is induced with this osmotic gradient, happening through the cells intracellularly through leaky tight junctions. A buildup of hydrostatic pressure results when accumulation of fluid in lateral spaces occurs. This causes water to flush into the interstitial fluid. So now we're in the blue area of the picture. The flow happens from the interstitial fluid from the lateral spaces and finally into the peritubular capillaries, which is our pink area seen in the picture. Due to the fact that plasma colloid osmotic pressure is greater in the peritubular capillaries, blood entering the peritubular capillaries where water is now located elevates the concentration of plasma proteins. This all can happen because of the extensive water filtration through the glomerular capillaries upstream. Concentrated into smaller volume of plasma, H2O plasma proteins that were left in the glomerulus cause an increase in pi-p of the unfiltered blood that exits the glomerulus and enters the peritubular capillaries. Water is then pulled into the peritubular capillaries due to this force, while at the same time hydrostatic pressure in the lateral spaces causes a push of water toward the capillaries. And this is just in fact where our 117 liters a day of reabsorbed water by the end of the proximal tubule come from. Now, moving on to urea absorption, which is indirectly correlated to active sodium reabsorption. Made from the breakdown of protein, urea is a waste product. This waste product has a passive reabsorption that is favored by a concentration gradient produced by an osmotically induced reabsorption of water in the proximal tubule to active sodium reabsorption. The amount of fluid remaining in the tubule gradually decreases due to extensive reabsorption of water from the proximal tubule. With this being said, some substances will be left behind, becoming more concentrated in tubular fluid, in our case being the most important, urea. As a filtered substance, the concentration of urea remains similar to that of its concentration in the plasma entering the peritubular capillaries. The concentration of urea becomes more concentrated in the adjacent capillaries than tubular fluid because of the quantity present at the end of the proximal tubule being almost threefold in the remaining filtered fluid. With this, a concentration gradient for urea is created in order for it to passively diffuse from the tubular lumen to the peritubular capillary plasma. Passive reabsorption of urea is only about 50% because of the somewhat permeable walls of the proximal tubules. Although elimination of filtered urea is only half, it is still an adequate amount being eliminated from the plasma, and each passing through nephrons, bringing us to the impaired kidney function, which results in elevated concentration of urea in the plasma. This is because less than half of the urea is removed. Not only this, but severe renal fluid was also associated with patients that had elevated urea levels. As a result, BUN, which is an acronym for blood, urea, and nitrogen, is now used as a measurement to assess kidney function. Okay, so to end the summary on urea reabsorption, I wanted to point out that the most serious consequence of renal failure is not pointed to the retention of urea. This is because it's not particularly toxic on its own, but it's rather due to the inadequate secretion of hydrogen and potassium. Okay, so now moving on to our clinical significance, which in this case is chronic kidney disease. In our study, there is a focus on patients with chronic kidney disease evaluating their levels of salivary creatinine and urea compared to those of a healthy individual. It involved taking 50 patients with chronic kidney disease in the last stages and 49 healthy individuals. Saliva and blood samples were taken from the patients in order to complete the analysis. Results showed elevated levels of salivary creatinine and urea in patients with chronic kidney disease. And not only this, there was also positive correlation between all three, meaning the blood, salivary creatinine, and urea. I also put the charts in there so you could see the numbers. The bottom table is plasma and salivary creatinine and urea levels in patients with chronic disease and the healthy controls. The top table is a correlation between plasma and salivary creatinine and urea levels in patients with CKD, and healthy controls. So in conclusion, to leave it short, analyzing a patient's salivary creatinine and urea can be used in patients with CKD in order to get their levels in blood. With this being said, amazingly, these two things can be used as a diagnostic biomarker for chronic kidney disease. If y'all want to look further into that study, the top resource is the one for that study. And that's all my resources. I will see you guys in the next one. Thank you.